Hi, my name is Lily Khang, and this is my biology role model project. So the biologist that I chose for this project is Sarah Dorr. Sarah Dorr is an assistant professor at the University of Florida. Sarah received a BA at New College of Florida in the Department of Biology in 2007. In 2015, she received her PhD at the University of Arizona in the Department of Plant Pathology and Microbiology. And in 2020, Sarah received her postdoctoral at Michigan State University in the Department of Biochemistry and Molecular Biology. Sarah is currently in the virology department and is currently studying how bacterioviruses interact with their hosts. While I was conducting my research, I was unable to find anything about Sarah's personal interests, nor the reason why she got into science, but I was able to find on Rate My Professor of how her students viewed her classes. A lot of her students stated that she was really good at explaining concepts and that she was very approachable and kind. This shows a lot of Sarah's character. So why I chose Sarah? I chose Sarah because she and I have a connection through bacteriophage research. In my sophomore year of high school, I was given the opportunity to also conduct research on bacteriophage with antibiotic-resistant E. coli and Pseudomonas through the University of Montana. While I'm not interested in microbiology as a major, this has influenced my interest in research. Bacteriophage research is interesting, and I believe it has a big impact on the future, especially with the increase of antibiotic resistance. So Sarah's most recent research was published in 2025, and the title of this paper was The Structure of Shigella Virus SF14 Reviews the Presence of 2 decoration proteins and 2 long-tail fibers. The purpose of this overall research was to find the structures of SF14, which is a bacteriophage that infects the human pathogen Shigella flexneri. The background of this is that there was a low-resolution image of SF14 which showed that there were proteins on its capsid. The capsid of a virus is basically just its protective shell. So the methods for this research is 1, to cultivate SF14. To cultivate SF14, the bacteriophage was grown in LB solution and then poured onto a plate. After the phage were collected from the plate, they were plunged into frozen liquid ethane and then read by cryo-SPARC. Cryo-SPARC is software that can create 3D structures of molecules. After, the images were put into AlphaFold to help determine individual structures of the proteins. AlphaFold is an AI system that determines the 3D structures through amino acids. From the methods, the results and discussion, 2 types of proteins were found. The bee tulip lattice and the hawk-like were the proteins that were found. Whiskers and tails were also found for SF14. Understanding the structure of SF14 allows our scientists and researchers to determine its functions. This figure is the figure shown in the paper. This is basically a 3D model of the SF14 virus. The circular head at the top is the capsid, and the longer bottom area is the tail. The figure represents the overall structure of SF14 and gives a visual representation of the head and tail that were displayed. The overall colors shown of the SF14 is basically the number of angstroms in the bacteriophage. There are bars on the sides that determine the different numbers of angstroms in each area based on the color coordination. Thank you for listening to my presentation. I really do find bacteriophage research very interesting, and it's really nice to find a biologist who's also doing it as well. Sarah's also a woman, so that's also really nice to have in terms of research. And she's definitely someone who I look up to. Reading more about bacteriophage really got my interest sparked again, and I'm really excited to learn more about them in the future. And then these are my citations.